On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, NanoTemper, I'd like to welcome you to Formulation and Stability Screening of Biotherapeutics. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now, I would like to introduce our moderator for today. He is Charles Heffern, Product Manager with NanoTemper. Welcome, Charles. The webinar is yours.
Thank you, Elizabeth. And hello, everyone, and thanks for joining us today for this presentation on nano-differential scanning fluorimetry and its applications by Jie Wen. Before Jie begins, I'd like to provide a brief introduction for anyone who is unfamiliar with Jie or his work. Jie is a senior scientist at Amgen, where he has been working in biophysical characterization and higher order structure since 1993. In his 27 years of experience at Amgen, Jie has experienced firsthand the evolving landscape of biophysical characterization and biologic development and characterization. Over this period, his work has resulted in more than 30 publications. With research interests spanning higher order structure, protein stability, and biophysical characterization of proteins, protein-protein interactions, and protein-small molecule interactions using a plethora of techniques, including nanoDSF, DSC, ITC, amongst other biophysical techniques, Jie has a real wealth of experience in biologic development. So, without any further ado, I am very much looking forward to this presentation, and please take it away, Jie.
Thank you, Charles, for the introduction. Today, I'm going to talk about nano-differential scanning fluorimetry and its applications. Take a look at part 1, introduction. Before I show you nanoDSF, I would like to mention the current industrial standard, differential scanning calorimetry, DSC, and its limitations. For DSC, the protein concentration used are typically around 0.2 to 2 mg per ml. The protein sample consumption is 400 microliter each sample. The throughput is 1 sample per injection. So we hope we can have a method that offers a wider concentration range, lower consumption of material, and a higher throughput. Now let's take a look at nanoDSF. As you know, a protein with a tryptophan shows intrinsic fluorescence. The top trace in the left figure is the fluorescence spectrum of a folded protein. When a protein unfolds, the fluorescence intensity becomes lower, and the peak may have red or blue shift. If we plot the fluorescence intensity ratio, 350 versus 330 nanometer, and the function of temperature, we can get the unfolding profile of this protein. On this unfolding profile, we can get the TN info. Now let's take a look at the sample consumption. For DSC, it uses 400 microliter per sample. For nanoDSF, it uses 10 microliter per sample. So nanoDSF requires considerably less amount of protein material. Now let's take a look at the throughput. For DSC, it can run 1 sample about 2 hours. For nanoDSF, it can run 48 samples about 2 hours. The high throughput of nanoDSF is achieved by the rapid-on-the-fly mode for nearly simultaneous measurements of 48 samples in the capillary and each temperature point. So nanoDSF provides significantly higher throughput for the analysis of the therapeutic proteins. In this COVID-19 pandemic, we know one thing for sure, that is the throughput and the turnaround time of testing COVID-19 is so critical. Higher throughput and a quick turnaround time could be a matter of life or death. Now let's take a look at the concentration range. For DSC, the typical protein concentration range is 0.2 to 2 mg per ml. For nanoDSF, the typical protein concentration range is 0.05 to 200 mg per ml. NanoDSF can measure a wide concentration range of therapeutic proteins. To evaluate nanoDSF for thermostability analysis of therapeutic proteins, we selected 7 different maps, 4 different bite molecules, 2 different cytokines under different concentration. The left figure is a map. The right figure is a bite molecule. The black trace in the figures are the formulation buffers. The blue trace are the map or the bite molecules. From these 2 figures, you can see there are no transition observed for the formulation buffers. However, there are 2 thermal transition observed for the map and 1 thermal transition observed for the bite molecule. So in general, nanoDSF is able to detect thermal transition signals for map, bite molecules, and cytokines. Here is the repeatability of TM measurements of a map. We did 7 replicates. The repeatability for TM1 is 0.2 degrees Celsius. The repeatability for TM2 is 0.1 degrees Celsius. So the nanoDSF can measure the TM value of maps with higher precision. Here is the repeatability of TM measurements of bite molecules. We also did 7 replicates. The repeatability for the TM measurements is 0.1 degrees Celsius. So the nanoDSF can measure the TM value of bite molecules with high precision. Here is the comparison of nanoDSF and the DSF profile. In this figure, the blue trace is the nanoDSF profile. The black trace is the DSC profile. Here are the nanoDSF and the DSC TM values. From this figure, you can see nanoDSF and DSC show similar thermostability profiles for the maps. You may notice that there are some difference between the nanoDSF and the DSC TM value. This may be due to 2 factors: the different attributes that the 2 instruments measure, and the different instrumental heating environments. Because of the time difference, we recommend the side-by-side measurements use the same instrument, either nanoDSF or DSC, to study one set of samples. Here is the comparison of nanoDSF and the DSC profile. In this figure, blue trace is a nanoDSF profile. The black trace is DSC profile. Here are the nanoDSF and DSC team values. From this figure, once again, you can see nanoDSF and DSC shows similar thermostability profiles for bite molecules. Now let's switch gears and talk about part 2, nanoDSF applications. The first one is developability and formulation study. The second one is comparability study. Now let's take a look at the first one, developability and formulation study of therapeutic proteins. Before I show you the application, I would like to mention, since DSC and nanoDSF often show similar thermostability results, the data obtained by DSC can often be performed by nanoDSF. Therefore, nanoDSF can be used for developability and formulation studies of therapeutic proteins in a similar manner as DSC. Now let's take a look at the first study. In this developability study, the evaluation of TN is one of the key aspects. This study was published in the Journal of Biological Chemistry in 2017. The goal of this study was to engineer an IgG1 scaffold lacking effector functions with optimized developability. Scaffold 1.1, 1.2, 1.3 are the asparagine 297 mutants of IgG1 molecules. These asparagine 297 mutants result in no glycosylation and therefore loss of effector function. However, from this figure, you can see CEF 1.1, 1.2, and 1.3 has lower thermostability in CH2 domain. In order to improve the thermostability of these CF molecule, an intra-CH2 domain disulfide was introduced to CF 1.1. This modification made CF 2.0. As you see in this figure, the CH2 domain thermostability of CF 2.0 was improved significantly compared to CF 1.1. Although this study was previously performed by DSC, nanoDSF can also be used for such a developability study. Here is the formulation study. In this formulation study, the therapeutic protein candidate was evaluated under different formulation conditions. pH 5.0 buffer, pH 5.0 buffer with sodium chloride, pH 5.0 buffer with ammonium sulfate. So from this figure, you can see pH 5.0 buffers is the best buffer among the 3 buffers studied. Once again, although this study was previously performed by DSC, nanoDSF can also be used for such a formulation study and provide critical information. Here is the sample consumption and throughput advantage of nanoDSF in developability and formulation study. For protein sample consumption, DSC used 400 microliter. NanoDSF used 10 microliter per sample. The throughput, DSC can run 1 sample about 2 hours. NanoDSF can run 48 samples about 2 hours. So nanoDSF has significant advantages in developability and formulation studies in terms of sample consumption and throughput. Now let's take a look at the concentration range advantage of nanoDSF in developability and formulation studies. For DSC, the typical protein concentration range is 0.2 to 2 mg per ml. For nanoDSF, the typical protein concentration range is 0.05 to 200 mg per ml. The nanoDSF method enables the measurement of the thermostability attribute and both very high and very low concentrations of therapeutic proteins. Now let's take a look at another nanoDSF application. The comparability study for therapeutic proteins. In a comparability study, we do not compare nanoDSF and DSC anymore. In a comparability study, we use the same instrument, either nanoDSF or DSC, to study and compare the pre-change and post-change materials made by different processes. PRICE guideline Q5E changes in higher order structure can impact quality, stability, safety, and efficacy of biopharmaceutical products. We used 7 different maps, 4 different guide molecules, and 2 different cytokines to study the specificity and the precision of the nanoDSF method. The specificity of nanoDSF is defined as the ability of the method to measure protein signals accurately in the sample solution compared to the buffer. The method precision is measured by assessing its repeatability and is estimated as the square root of the residual variance. The equivalence acceptance criteria for the comparability study was established using the universal repeatability of the nanoDSF method and was defined as 3 times of the repeatability. The result was published in Analytical Biochemistry this year. Here I will show you an example of a comparability study. In this comparability study, the map was initially manufactured at Site 1 and later transferred to Site 2. The pre-change lots was from Site 1 and the post-change lots was from Site 2. 6 measurements were collected from each process. Here are nanoDSF thermograms. The blue trace are the post-change lots. The red trace are the pre-change lots. So the nanoDSF profiles are virtually similar for the pre-change and post-change lots. Here are the nanoDSF TN results. You can see pre-change lots here and the post-change lots here. They replicate and the TN values. We used JUMP and did the statistical analysis for these TN values. Here is a graphic summary of nanoDSF TN1 equivalence results. If you take a look at the left figure, the EAC limits are shown by the red horizontal traces. Here is the EMC limit. The confidence interval for the mean difference is shown by the blank trace here. So the two one-sided 95% confidence bounds, this one and this one, and the difference between the mean value here of the pre-change and the post-change lots for entirely within the EAC limit. Here is the graphical summary of nanoDSF TN2 equivalence results. Basically, the similar TN2 results were obtained. So the statistical analysis results can be also shown in a tabular format. Here is TN1, TN2, pre-change mean, post-change mean, difference in means, lower 95% confidence bound on the difference in means, and upper 95% confidence bound on the difference in means. And here is EAC. So here is the conclusion. So for this comparability study, the overall conclusion is The thermostability of the pre-change and post-change knots is comparable. Here is the summary. I have shown you the nanoDSF and its applications. NanoDSF has emerged as a powerful tool for thermostability analysis of therapeutic proteins. The nanoDSF method enables thermostability analysis over much wider concentration range, consumes considerably less materials, and provides significantly higher throughput. The nanoDSF method can be applied to many applications, such as developability and formulation study, as well as comparability studies of therapeutic proteins. Last but not least, I really want to thank these great people and Amgen for their contribution to this presentation. Matt, Nick, Harrison, Sheila, Fred, Yeta. I also want to thank Amgen Higher Order Structure Group, Amgen Biological, Relevance and Characterization Group, Amgen Attribute Sciences Department. Thank you for your attention.
Thank you, Jie, for that excellent presentation on how the Prometheus and the nanoDSF technology are enabling your work and research at Amgen, and how they are helping you to get better therapeutics into the clinic faster and into the lives of patients so they can make a difference. So at this point, I just want to do a quick wrap-up of focusing on exactly what this Prometheus instrument is and what the nanoDSF technology is, and how they are enabling the type of work that has been featured here by Jie from his case studies and recent publications that he has put out there on how they are helping his biologic characterization. From a high level, the Prometheus, why people are saying that it is this gold standard for precisely characterizing protein stability is that it is able to provide these precise measurements that Jie was featuring from his comparability studies. Moreover, this is done with a label-free measurement, meaning that samples can be taken in their native state without the addition of any dyes or any additional reagents, so it really facilitates assay setup. All of this can be done while consuming a very small amount of sample, and of course, as Jie was featuring for his comparability studies, this works with any concentration of protein, so no matter where you are in the workflow, whether it is that late-stage comparability assessment or even very early-stage discovery or still, let's say, mid-stage protein engineering efforts, the protein concentrations that you have that are appropriate for your characterization work with this platform and technology. When performing these measurements on the Prometheus, the way that it is able to, what it provides for you, no matter what environment you are in, are the, let's say, these same features and benefits here, right? With the high quality data that will help to inform the decisions that are being made about the development of various biologics and various pipeline decisions, as well as a fast time to being able to achieve these results, as Jie was featuring about the throughput in comparison to the previous standard that the lab would rely upon for most of their measurements, as well as a walk-up, easy-to-use platform that really anybody in the lab can walk up and both use the instrument as well as use the software to analyze and understand the data and the results. That makes the workflow much more simple and easy to execute. So I was going to touch briefly upon how the technology actually works and how it facilitates the type of work that Jie was just featuring in his presentation, just for a brief moment. So as Jie also spoke to at the very beginning of his presentation, The measurement that is performed on the Prometheus, this nanoDSF measurement, is a label-free measurement of the intrinsic fluorescence of a protein that can monitor the change in conformation of a protein from a folded to an unfolded state when it is being stressed in some way, whether it be from a thermal stress, let's say we were putting it through a thermal ramp, or in the presence of a chemical denaturant, right? Or even other physical stresses, such as, let's say, UV stresses, pH stresses, and other stresses that you might be interested in understanding how do your proteins respond to them. What this measurement is looking at in particular is the change in the fluorescence profile of those aromatic amino acids, predominantly tryptophan and tyrosine, as the protein conformation changes, as shown in this cartoon at the top. I think I can turn on the laser pointer here. Let's see if I can get this. Yes. Okay. So as can be seen in the cartoon, as this protein's conformation changes from the folded to the unfolded state, the environment that these aromatic amino acids, the tryptophan and tyrosine, are in, evolves and changes as that protein loses its structure. And this changes that fluorescence profile, as shown in the figure that Jie presented at the beginning of his presentation. And it is this change in that fluorescence profile that is monitored on the Prometheus to help identify and ascertain the conformational stability. Simultaneous to that conformational measurement, this is something that wasn't featured in Jie's presentation, there's also the ability to identify the formation of aggregates in a sample. So here, with the formation of aggregates, what we are looking at, we are identifying this through changes to the sample turbidity that are being measured with our back reflection technology. Here, what we can see in this cartoon is a little cross-section profile of the sample holders from which the measurement is being made. So here we are looking down the barrel of a capillary. And here, looking down the barrel of this capillary, we can see that essentially from above is where all of our optical setup is located. Here we are passing light through the sample in the near UV, and in the presence of aggregates, there is attenuation of the light that is being passed through, and we can use this attenuation to identify the onset and extent of the aggregation of a sample. It is this simultaneous measurement of these two different properties that enables the Prometheus to be able to make the impact that it's having on the work that is featured by Jie and is being done in other biotech and biopharma companies and organizations, to be able to identify what is anything from a lead candidate molecule to the optimal formulation or engineering construct of a molecule, or even to identify what are the effects of manufacturing or processing changes for clinical molecules that are in the clinic, to really provide that confidence for the regulatory bodies that there are no changes to the higher order structure when those changes occur. Here, I'd like to quickly dig into a little bit of specifics about the instrument design and how the design of the instrument has really built up to enable and facilitate this high quality characterization of biologics and biotherapeutics. So in brief, right, so all of the samples are, as Jie mentioned, loaded up in these sample holders, these capillaries, which are placed directly on a thermal element, this Peltier, that can be ramped anywhere from 15 to 95 degrees, that can be upgraded to potentially reach the higher temperatures for more thermally stable molecules all the way up to 110 degrees Celsius. It is this direct contact between the capillaries and the thermal element that really facilitates an optimal thermal transfer for a very precise TM, as well as those very small air bars and that high level of reproducibility that Jie was featuring. Moreover, these capillaries have a small diameter, which helps facilitate that broad dynamic range, that broad range of concentrations that can be measured on the platform. Jie was showing from 50 micrograms to 100 milligrams per mil in his work. Of course, the Prometheus, the nanoDSF technology can go even beyond those limits of detection, all the way down to single-digit micrograms per milliliter concentration, or in excess of multi-hundreds of mgs per mil of concentration. And this small inner diameter also helps with the thermal properties for that fast transfer of heat and temperature into the samples for that very precise and accurate measurement. Moreover, these sample holders provide a very, let's say, optimal workflow because of the way that they are designed. There is no sealing necessary. So all that needs to happen is that the samples are loaded into the capillaries, placed on the thermal element, and then they are ready to measure. The measurement itself is performed using the detectors and optics for these back reflection and fluorescence measurements that are using precise LEDs for the excitation of the fluorescence and passing the near UV light for the back reflection, as well as photomultiplier tubes to detect the light that is coming out for the highest quality fluorescence measurements. And this is what enables the measurement of that intrinsic fluorescence at 330 and 350 nanometers to look at the redshifts that are occurring when proteins unfold, as well as the back reflection in the near UV. So one thing that Jie featured heavily in his presentation was how the Prometheus has this level of specificity, accuracy, and precision that his work requires for his group, right? And that's something that we feature right here in an application note as well, or a tech note about the precision and accuracy of the instrument. And it is all of those features that we were just talking about, the design of the instrument, that combine to be able to determine those inflection points with a standard deviation or an error bar of less than 1/10 of a degree Celsius, as well as identify the fluorescence profile to really detect even the most subtle transitions within 0.008. So we can have a high degree of confidence. One additional part of this repeatability, accuracy, and precision is also the intermediate precision, or the precision between different instruments. So let's say that instead of just one lab working with an instrument, you are part of a group that has to work with other collaborators who are either upstream or downstream from you in the workflow of developing a biologic, or you might have external partners who are either at a CRO or at another company that you are collaborating with on the development of a molecule. Of course, you want to ensure that the results that are achieved in your lab are similar and relatable and comparable to the results that are achieved in your collaborator, your partner's lab as well, right? And this is where the intermediate precision comes in. So here, if we look at the difference between 10 different instruments, that standard deviation, the confidence of the TMs, is still within 0.3 of a degree Celsius, and that initial ratio, which is used for detecting the conformational stability and the unfolding of a molecule, are within 0.02. Of course, the Prometheus has applications beyond just determining the thermal stability of a biologic or a molecule. As briefly mentioned earlier, these expand over multiple applications, such as looking at chemical unfolding measurements, where you might be adding some sort of chaotrope to unfold a molecule, determination of delta G, or those self-interactions through changes in the chemical unfolding as a function of the concentration of the biologic or the protein of interest, and of course, various other applications when it comes to optimizing assays or buffer conditions that are being used for an antigen of interest or some other protein that might be part of your drug discovery workflow, right? These are all things that the Prometheus can help to understand and facilitate that part of the work. So I'm not going to really touch upon any of this here, but I wanted to briefly sort of just bring up that in addition to this late stage characterization that Jie has been featuring so nicely here, of course, there are many other publications out there where groups are talking about how these same properties of the Prometheus and nanoDSF technology that are so important to Jie, that he so nicely lays out in his presentation about that specificity, sensitivity, accuracy, precision, limited detection, and resolution, really enable his work in the late stage characterization, those comparability studies. Those same properties were critically important for even early stage discovery and early stage work at groups like Merck, where there's this publication on predicting the biologic developability profiles through early stage screening. By all means, you can go online and find this publication right here through this VOI, or in, let's say, not quite as early stage, but we're performing some sort of protein engineering work, which actually Jie featured quite nice in his presentation as well, about looking at those different constructs of the different protein, of the different mutants in there, right? So here is a publication from this year as well, from a group at Janssen, talking about how they are engineering in some stability enhancements of this transgenic animal-derived antibody and how these same instrument properties that Jie was featuring previously really impact their work and help to facilitate and enable that. And of course, if you're interested, there is the DOI for that publication as well. Finally, Jie was featuring most of his work that is done right here on our Prometheus NT48, which is our very flexible throughput platform. But of course, there are different formats of the Prometheus instrument that address various workflow and throughput needs. We also have a version of the Prometheus instrument that provides easy sample handling that works with direct loading from 384 well plates. And what's very fun and interesting about this instrument is that it actually has a turnkey implementation for full automation, where samples can be essentially set up to be loaded by a robotic autosampler, and this can measure through multiple plates of samples without any user intervention. So with that, I would like to thank you for your attention and for joining Jie and myself for this presentation today. And we will begin taking the questions that have been coming in throughout the presentation, and we will start the question and answer session.